Good afternoon or morning, everybody. This is Chapter 1, Microbiology, from our textbook by Jeffrey Palmerville in the Fundamentals of Microbiology. So this is your very first lecture. All of our lectures will be online and posted for you, and then your job is to listen to each of these lectures prior to class. All right? So the first thing I want to go over before we get into the lecture material is where you're going to find all the information. If you see here, I post all of my lecture notes on Canvas. I will also post all of my links to my actual lectures on Canvas. So if you click on this link here, it brings you to Spoon River College website. Hold on, it's got one. There you are. If you go to the Canvas link here, you're going to be brought to your home page. If you're already signed in, it'll go right to your home page. If you're not, then you're going to have to sign in. And if you're struggling with signing in, you can call the help desk or you can just talk to me, and we'll be meeting tomorrow evening. So here is your class. This is what you will see. And if you want to find your syllabus, you can go, just click here. But I'm going to go right to the course material here. And here we're on all the modules. So here's unknown poster project information. Here's your first chapter 1 lecture PowerPoint. So once I have this lecture posted or finished, I'll post it on YouTube, and then I'll post the link for you here as well, okay? So I'm going to now go back to this. Nope, I lied. Go back to this. Here's some more information. My name, Krista Winters. Where my office is, it's in Canton in room T208. In Macomb, I'm typically found in the LRC or in the laboratory. And then here's my email address. You have a link directly to it. So let's get into lecture 1. Chapter 1 is all about the history of microbiology. So what is microbiology? When did we start studying microbiology? How old are microorganisms? What made us decide to study microbiology? So the first thing you need to know is microbiology is the study of microscopic organisms or microscopic structures. And microorganisms are found all over the earth. They're the foundation of all life on earth. They've been around for about 3.5 billion years. So not as old as the earth, but they're really, really old. And you can see where, so here, where earth is formed. And then we have the first signs of life right here. And it's way up here that humans come to be part of this world. So we're studying these microscopic, very archaic organisms in this class. Super fun, right? So how did microbiology come to be? Well, microbiology, in a nutshell, is the study of microbes. And before we could study microbes, we had to be able to see microbes. So that we could figure out tools to study microbes. So we made microscopes, things that could see structures that were too small for our eyeballs to see. So some of the first things that people probably used were things like magnifying glasses. And eventually they were like, wait, if I can use this magnifying glass, I wonder what would happen if I add one magnifying glass to another magnifying glass in a certain way, allow light to move through, and they made their first microscopes. Once we started seeing organisms that were microscopic, we started learning techniques to culture these organisms so we could grow them, and then we could learn more about them. And now we actually can look at their genetics, so the genes that make them who they are. the nucleotides that are found in all living things, which is really exciting. So who was one of the first people to actually see microorganisms? Robert Hooke in the 1600s actually produced one of the first microscopes, and he looked at structures. First of all, he looked at cork, and he noticed that cork had little tiny cells, or what he called cells. They had little structures that looked like the same types of cells that were in the monastery. And he coined the term cell by because they looked very similar to the cells in the monastery. He published his first drawings in the Micrographia, and the first drawings were what he called microscopic flowers. And so here he had these microscopic plants or flowers, and what he found out they were actually mushrooms. Then we, a little bit later, we get Antoine von Leeuwenhoek. He was a Dutch drapist. This guy was amazing. He was an excellent worker. He made some of the finest drapes in the area, and he made a lot of money on his drapes because rich individuals would buy them. But the reason he was able to make such perfect drapes was because he used this tiny little magnifying microscope that could magnify 200 times more or larger than what he could see normally. And so he would use that microscope to look at the seams of his drapes to make sure that every single stitch was perfect. And one day he just decided, hey, I wonder what would happen if I looked at pond water or lake water under my microscope. I wonder what would happen. And so he looked, and what he noticed were there's these little things that would zip around in the water. He couldn't really see them well because they would move so fast. And he was like, what in the world? These are tiny little animals. And he called them animalcules. So then he looked at teeth scrapings. He's like, well, I wonder, is there something, our teeth gets this grimy gunk on it. I wonder if there's anything in there. And he saw under the microscope the first bacteria. And so he is considered the father of microbiology by many. So not everybody, but by many. He is considered the father of microbiology because he actually saw and drew the first bacteria. So then the question comes, where did these microbes come from? So prior to the 1600s, 1700s, a lot of individuals, scientists, the church goers, church clergy, thought that life came from non-life, that things just were produced or came about spontaneously, often associated with how good of a Christian you were. If you were a bad Christian, then you may get diseased, get very sick, and die. If you're a good Christian, maybe you will get pregnant. Maybe God will bless you with a pregnancy spontaneously. And so this was the theory. And in general, a theory is something that people believe is true. So in biology, a theory is regarded as the truth. a theory is something, or started out as a hypothesis that was tested and was supported, and it was supported multiple times. And when you have a hypothesis supported by multiple individuals running the same tests and it's supported multiple times, then it becomes a theory. So things like the cell theory, the theory of relativity, the theory of evolution, those are all theories, right? Relativity is not a science, or it's not a biology theory, but it's a theory. In biology, we don't have laws. The reason we don't have laws is because living things are constantly changing. And so it's really hard to make a law. But biologists, they believe that theories are the truth. So if you have a theory, you pretty much are very confident that this is truth. But things can change. And we know that because, we used to think the earth was flat. And people who said the earth was round were laughed at, and now we know the earth is round. Well, back in the 15 and 1600s, people thought that life came from non-life. This was the theory of spontaneous generation. And spontaneous generation basically stated that if you take and put certain things together and leave them sit for a period of time, life will arise. One example, dirty clothing and wheat or other type of grain. You put that in a dark cellar for a few weeks, and mice will all of a sudden appear. So that's how you make mice. And that sounds really silly now, and we know it's not correct. But back then, people believed this. And because they believed it, it's hard to make someone go against the comfortable, very well thought out belief. And so people who did not agree with spontaneous generation, these were called, these individuals were going against the norm. And so Leeuwenhoek suggested that maggots, so flies, produce eggs that when they hatch, the organism that comes out is called a maggot. It's a little worm. He said that maggots probably came from eggs that were laid into decaying meat. People were like, nah. So Francisco Redi said, well, let's do an experiment. And he did. He took and put raw meat into two different containers. One he covered with this stuff called cheesecloth. The other one was uncovered. Both of them had rotten meat, but only the open jar actually produced maggots. And so he said, hey, this supports the Leeuwenhoek's hypothesis that maggots come from flies that lay eggs, because flies got onto the meat, and they probably laid eggs, and that's where the maggots came. With the one with the cheesecloth, no flies could land on the meat, and so no eggs were produced. I'm going to stop here because these videos can only be 15 minutes long, and I'm at 14:27, and because of that, I'm going to stop so that I don't get cut off, because sometimes when I get going, I lose track of where I'm at, and the next thing I know, I have, well, the video gets cut off on me. So I'll see you in our next video. Bye.